I'm Cristina López Vicario, postdoctoral researcher of the Group of Inflammation and Liver Disease at the IDIBAPS institution, which is associated to the Hospital Clínic of Barcelona. And together with Dr. Joan Clària, we are performing an investigation which is focused on the identification of new lipid mediators of inflammation in the circulation of patients with acutely decompensation cirrhosis. These patients eventually present extrahepatic organ failures. They also present high risk of short-term mortality, and this is a condition that is also known as acute on chronic liver failure or ACLF.
I'm Brian Contreras, and I'm a PhD candidate in the research group of Professor Joan Clària. And in this project, we found that the patients with acute decompensated cirrhosis presented high levels of a lipid mediator, namely 910-DHOMB, which is a lipid mediator derived from the polyunsaturated fatty acid linoleic acid, which is related to pro-inflammatory processes. We hypothesized that this lipid mediator, 910-DHOMB, might be the responsible of the increased risk of these patients to present recurrent and bacterial infections. To prove our hypothesis, we performed different bioassays with different cells of the immune system in presence of this lipid mediator. We found out that this 910-DHOMB leukotoxin was able to impair key immune defensive functions in polymorphonuclear leukocytes, such as respiratory bars, degranulation, and phagocytosis. We also wanted to see if this 910-DHOMB would also affect other cells of the immune system, mononuclear leukocytes. And we observed that 910-DHOMB was able to impair the cytokine production of these mononuclear leukocytes, and we think that it also plays a role in impairing the mitochondrial dynamics.
So our results suggest that high levels of the leukotoxin 910-DHOMB in patients with ACLF worsens their susceptibility to infection, and also they are more prone to precipitating events. From this research, we are looking for new biomarkers. So this way, we could have a target and a treatment to improve the life quality of patients with cirrhosis. So taken together, our results suggest that inhibiting the production of this 910-DHOMB, which is also known as a leukotoxin, we would improve these patients, and we could also improve the prognosis.